Hey everyone, thank you for joining me today for episode 759 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am joined by our guest, Dr. John Majercak, to talk about life and science. Listeners' support for this episode was provided by Lampire Biological Laboratories. I hope you enjoy getting a behind-the-scenes look into the life and experiences of another researcher in today's episode. And if you want to meet even more of today's scientists, you can check out our website at peoplebehindthescience.com or subscribe to our show on your favorite podcast platform. And today, John is going to share some of his insights and stories. So listeners, get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. John Majercak. So, John, welcome to our show today. How are you?
Very good. How are you, Marie?
I'm doing quite well. Thanks for asking. And we are excited to learn more about you and the work that you do. But let me first start by telling our listeners a little bit more about your current position and how you got there. So listeners, John is Head of Antibody Discovery at Lampire Biological Laboratories, a life science company at the forefront of innovation and discovery. John received his PhD in biochemistry from Rutgers University, and afterwards he completed an industry postdoctoral fellowship at Novartis. Over the next nearly 2 decades, John worked at several companies, rising in the ranks to hold various positions at Merck, Boehringer Ingelheim, Johnson & Johnson, Bristol Myers Squibb, and Molecular Templates, before joining the team at Lampire in 2023. And John, today we are excited to get to know you both as a scientist, of course, but also as a person. So can you tell us, what do you like to do when you're not doing science?
So when I'm not doing science, I do like to explore different sciences outside of my main science, which is really genetics and biochemistry and antibody discovery work. I really love physics and space exploration and astronomy related activities. So I listen to a lot of podcasts, and I have several ex-colleagues that actually have moved over into this space. Beyond that, I love to do home renovations, believe it or not.
Ooh.
My family and I, we've lived in several locations on the East Coast, and we've owned several older homes, historic homes, I guess, in a way. And I've spent a lot of time over the years learning to do a lot of the carpentry and the electrical work and the plumbing and what it takes to kind of bring these older homes back to life.
Oh, I think that's so cool. Do you have any ongoing projects right now in your current home?
Yeah, we do. We have a 3rd floor bathroom in mid-reno right now, but there's always something going on.
Oh, absolutely. I had an HGTV phase where we were always trying to renovate something, it seemed like. So I know how that goes. Well, you mentioned that in addition to your current science, you like to explore other disciplines. I'd love to talk about your current science in a little bit more detail. So how do you describe the work that you do right now to someone who's not in science or perhaps is outside of your field?
The company I'm with now, Lampire Biological Laboratories, is really, let's say, a blood products company. And honestly, the majority of the work is antibodies that are in the blood. And antibodies are circulating through everyone's bodies, humans and all animals. So the work that we do is focus on those antibodies that are circulating in the blood. We basically generate new antibodies or provide companies or academic institutions or biotechs with the existing antibodies that are in the blood. A lot of my work is focused on the custom production of these antibodies. So if you're not familiar with the composition of blood, there's red blood cells, there's white blood cells, and the white blood cells are the specialized cells that are involved in your immune response, and they're the ones that generate antibodies. My focus really is to focus on those white blood cells that produce the antibodies and capture the genetic components that make the antibodies that are circulating in your blood. So these antibodies are used across the industry, and very importantly, they're used for antibody therapeutics, many of which are on the market now for a lot of oncologic and immune-related diseases and disorders.
Definitely. And I think, John, it can be really exciting to work in the biological and biomedical sciences fields where you can see that your work can have a really big impact. But I think some days you need a little extra motivation, a little inspiration to get you going. So do you have a favorite quote or a saying or something that just really keeps you motivated and inspired?
One thing that I've been known to say is to recognize your challenges, but also celebrate your victories.
So how do you incorporate that into your everyday life?
Yeah, I say that a lot because we face a lot of challenges. We work on a lot of projects that are very challenging for various reasons. So it's important to recognize those from a team perspective and kind of set expectations and to be able to effectively go after these and realizing that you're probably going to have some bumps in the road along the way and maybe have some learnings that you can apply to actually get to your goal. And then when you achieve a good result in the end, are able to deliver something that is impactful. Generally, we like to celebrate that a little bit, go out to lunch or that sort of thing.
Oh, definitely. And I think to make it through some of those bumps in the road along the way, as you put it, I think it can be helpful to have these mentors or these role models or these people that you work with to help you get through difficult times. So when you think about your career, John, are there particular people who rise to the top of your mind as mentors and role models who really helped you?
There is actually two people that were very instrumental for me as a young scientist. And this goes back a few years, so I'm going to date myself, but I did my graduate work at Rutgers. And actually, this was a program between Rutgers and the University of Dentistry of New Jersey, which is the medical school there. And I was in the lab of a Dr. Isaac Edery, and he actually joined why I was doing some, I guess it was like a part-time job that I was doing and basically working in lab services. So fixing glassware and providing pipette tips and filling pipette boxes. Right. And I wasn't really involved in the science, but this was sophomore, junior year of my undergrad. And Isaac showed up. He was a fruit fly investigator investigating circadian rhythms being day-night rhythms that are really encoded in our brains, more or less. And he brought me under his fold in a way where I started working a little bit in his lab, moving fruit flies back and forth. And I ended up continuing some undergraduate research in his lab, ultimately did my graduate work in Isaac's lab. And he was the type of person who was incredibly smart, very solid scientifically, realized that there's always a learning curve. And we certainly had those learning curves. And so we failed a lot, but then we succeeded a lot. And lucky enough, we were able to put out some nice publications. And he was a fantastic mentor. And recently, actually, he surprised me when I was in one of my more recent positions. And I was working at Molecular Templates out of Manhattan. And his daughter, who's now in the sciences, had suggested that she wanted to kind of just come by and, get some advice from me as far as, career mentorship. And she actually ended up showing up with Isaac, which was a great surprise. And I hadn't seen him in about 20 years, but he was a fantastic mentor during my graduate studies. Then the other person that kind of slingshot me into my industrial career was my postdoc advisor, Dahlia Cohen. Dahlia was the head of the functional genomics department at Novartis, originally in New Jersey, and then we had moved up to Cambridge, Massachusetts. She was a fantastic, hard-driving scientist. This was in the early days of the Human Genome Project, and she was involved in several aspects, and we were developing siRNA and cDNA kind of genomic screens. Again, this was in the early days of these genomics-based discoveries and making these libraries for drug target validation and identification. And Dahlia was instrumental in driving that department. Her department was split between Basel, Switzerland, and New Jersey or Cambridge, Massachusetts. And so one of her sayings was, there's 24 hours in a day, but sometimes there's more. Just to give you an idea of the type of person she was really dedicated to her craft and really motivated us.
Oh, I think that's wonderful. And I know you mentioned that already in undergraduate, you were getting your feet wet working in a laboratory. And I think that is a perfect way to expose people to kind of what science is really like. But what first turned your eye towards science and got you moving towards this path?
I really just loved being in the lab initially. I liked the exploration. And when we were working in the early days and looking at circadian rhythms, this was still a young field and we were identifying the early molecular components that were driving and underlying the circadian rhythms in animals. And just learning that these small insects actually were an entree into understanding human behavior ultimately. It's pretty wild if you think about it. There's that translation, even from an insect all the way to humans, and being able to study. And we made some seminal discoveries in how the molecular mechanisms inside the brain of these fruit flies were driving and regulating circadian rhythms and other sort of behavioral outputs in the animals. And these were found to translate to mammals as well as to humans. So kind of getting my feet wet in that really cemented my career in the sciences.
I think that's awesome. So can you give us maybe a summary of some of these key points along your career path that really helped you, John, get to where you are today there at Lampire?
I've had a long career. And initially I was very much focused on neurological disorders, Alzheimer's disease primarily, other neuropsychiatric as well as neuropathic pain related work. And this was work that I had initiated at Novartis, but then joined a very productive Alzheimer's department at Merck. And that was the first time that I was able to really follow a project to almost the clinic. It didn't quite make it to the clinic. So being able to see how one identifies, let's say, a novel drug target. This was a target that was potentially involved in Alzheimer's disease, and being able to follow that project through the discovery phase where we're finding small molecule inhibitors against this target that were able to cross the blood-brain barrier and had some effects in vivo, and then to try to translate that and potentially test that in humans. Unfortunately, that particular target did not make it into clinical trials, as do a lot of these concepts that might have that early potential to be a therapeutic intervention, a small molecule or an antibody therapeutic, kind of fail in those early stages and just maybe don't quite meet the endpoints that you want them to meet. At that point during my career at Merck, what really got me into antibody discovery, and I'm fully into, there's kind of like, I would say three, maybe even more now, but three major therapeutic modalities in the industry, at least in my industry. One of them being small molecules, the other one being vaccines, and the third one being biologics or antibody therapeutics. So at that time, I got my first taste of biologics and was still at Merck. And we started to do some collaborative activities with a company called Ablinks. Ablinks was a pioneer in the field of the single domain antibodies, or what they call nanobodies, something that I still work on extensively today. And this really got me to move in the direction of biologics and to realize that that was really where a lot of my, let's say, strengths as a scientist laid. So that was a pivotal moment, having those early work and being able to do some due diligence around the nanobodies that we were working on in those early days at Merck. And then after that, moving into Boehringer Ingelheim, J&J, Bristol Myers, and ultimately Molecular Templates, and now Lampire Biologicals. There's many key projects that I was involved with, those different companies that I'm very proud of as well.
Fantastic. And John, can you tell us, why did you decide to make the jump and join the team there at Lampire?
As described, I do have a long history in pharma and biotech companies that has been fantastic. But Lampire works with basically all of the large pharmaceutical companies that you can imagine, as well as biotechs, as well as academic researchers. So after a long career within one company, and you have the ability to bring, let's say, some new technologies more broadly, make them available to a lot of different groups and teams and organizations, is really nice. It's really fantastic to be able to do that. And so Lampire, essentially being a CRO that partners and provides services across the research fields, both in industry and academic research fields, this has allowed me to do that here. Lampire, of course, is a company that has a long history working with companies, providing the blood products, providing polyclonal antibody approaches, which is a very big part of what we do. And the polyclonal approaches are still very much used for generating the reagents that are used in clinical assays. So let's say to look at anti-drug antibodies that are important to understanding the PK-PD relationships as the therapeutic is going through the clinic, as well as these polyclonal antibodies are used in establishing many other sort of assays in the field that academic and industry researchers use. But then more recently, again, to bring all that experience that Lampire has in the blood products and in the polyclonal, but also now in the monoclonal area, it's very exciting. So having been a part of a lot of different companies, I can say that this position at Lampire is really unique because I do have the opportunity to advance the science across many different areas of antibody and biologics discovery. And here at Lampire, we have a very experienced and talented team that I get to work with, both on the farm, in our small animal facilities, as well as the lab and the leadership here. It's truly a unique company as it still is one of these companies where it's family owned and it really has that sort of feel that we're all together working towards this one goal of delivering the highest quality products and science to our partners.
Oh, wonderful. I think you've done some phenomenal work over the years, and I'm sure it's exciting to be in Lampire in this area, kind of having control over their antibody discovery program as the leader of it. So can you give us an example of maybe a project that you're working on right now, John, that you are just the most excited about?
I mentioned nanobodies, and this is a project that, again, there's single domain antibodies that are expressed in camelids. So llamas, alpacas, camels, they express these special antibodies. They go by a few different names. Again, nanobodies is one of them. Single domain only antibodies is another one. VHHs is a third. But they're very unique in that they are smaller than your conventional antibody. Your conventional antibody, as it's expressed, let's say as it's floating around in your blood, is around 150 kilodaltons. A VHH, or a nanobody antibody, as it's expressed, is much smaller than that, 75 kilodaltons, but you can even make it smaller. So you can actually express just the binding domain of this heavy chain only antibody and produce what we call on its own a nanobody. The nanobody is very small, about 15 kilodaltons. And because of its size, primarily, it can achieve some special binding properties. One of them being it can reach these novel epitopes on our drug targets. And what I mean by that is it could be a drug target that's involved in cancer. Let's say something that is misexpressed on a cancer cell. And you might be able to find a conventional antibody that you might express in your body or another animal might express, and it might be able to bind that target. But it may not be able to hit every available binding site, or it might not be able to access every point of contact. These nanobodies, because of their size, can reach these novel epitopes and therefore allows us additional opportunities to go after these sorts of drug targets or create novel therapeutics. I guess I should also say, kind of related to nanobodies, is believe it or not, cows, bovine that is, cows, make an even smaller antibody that has, within the past several years, started to explore its therapeutic potential. And these are called picobodies. And these things, they're even smaller. So instead of nano, we call them picobodies, and they're about 3 to 5 kilodaltons, which is really small. 3 to 5 kilodaltons is like 30 to 50 amino acids. That's relatively small for a protein, but it folds up through a series of different disulfide bonds. And there's some flexibility, let's say, in that disulfide bonding, which allows it some additional conformational space to explore. And these picobodies are yet to be fully explored binding modality. But just like nanobodies, I think there's some real potential there, not only for a therapeutic, but also for the other diagnostic or assay-related reagents that one would use in the laboratory.
Interesting. So, John, do you currently work then with different partners or clients on discovering these nano or picobodies, or how does this process work?
So Lampire has a long history of working with llamas and alpacas that these nanobodies are derived from. But more recently, what we've done here is we've developed what we call a single B cell process that is applied to discovering or identifying these nanobodies. So how this works, this is monoclonal antibody discovery, more or less. A lot of people may have heard about hybridoma technology, and that's been a standard in the field for a long time. The challenge with hybridoma technology is that it's really species restricted. So it really only works with small animals like mice and rats and perhaps rabbits. But larger animals, it really doesn't work for larger animals. And so in recent years, there's been new modalities or new platforms developed to isolate monoclonal antibodies, or in this case, specifically nanobodies from llamas and alpacas. So what this looks like is essentially we draw blood from the animal, and this would be analogous to someone going into their local blood drive and taking their blood. And essentially, we take the animal's blood, and then we isolate what's called the buffy coat. In this buffy coat are the white blood cells. The white blood cells is a pretty good mix of different monocytes and neutrophils and eosinophils, importantly, lymphocytes. And lymphocytes are the cells that we want. And basically it's a subpopulation of what we call memory B cells that are in that lymphocyte lineage. So that's what we're targeting. And the way that we specifically isolate these antigen binding memory B cells is through a panel of phenotypic markers as well as fluorescently tagged target molecules. So these white blood cells in this mix are incubated or mixed with this panel of markers. And then we use a flow cytometry-based platform to specifically isolate these antigen-positive B cells. I should note that this significant investment has been made as far as, time and monies, I guess, from Lampire to develop this panel for llamas and alpacas, because these sorts of phenotypic panels for isolating antigen positive antibody expressing cells have been developed in the industry now for several years, primarily from mice and from humans and also from rabbits. But what we've done is we've generated these markers that they specifically can work in llamas and alpacas and specifically work for llamas and alpacas to discover these nanobodies. So following the sorting, basically what we call this FACS sorting or this flow cytometry-based sorting or isolation of these cells, then we go in and we use next generation sequencing to specifically amplify the VHH or the nanobody sequence from these cells. And that is then analyzed in what we call a repertoire sequencing to understand sequence families and how these sequences might relate to one another as far as affinity maturation events in vivo. And then we go through recombinant expression to express these monoclonal antibodies. These would be monoclonal antibodies at this time, specifically nanobodies that are monoclonal, and then we screen them. And as you can imagine, there's a whole host of different ways that we can screen these. And that itself is a huge field in how you screen these things and how you might look at kinetics, how tightly or how weakly these bind your target, how specific they bind your target, or do they have off-target binding, which is generally not good, not wanted. Or we can even get to the point where we're determining exactly what epitope or what particular amino acids on the target that we are binding. So there's a similar process also for the picobodies, and this works, and it's similar to the nanobodies, but it's not as robust just yet. We're still in the phase of building up those new markers, a new panel of markers to isolate specifically the antibodies in the cows that have these picobodies. But as it stands right now, we can certainly isolate these picobodies. And again, the approach is similar. The sequencing approach is a little bit different in that the picobody is more or less a subdomain of the antibody in the bovine. So our sequencing is targeted, very targeted sequencing to get those picobodies. The nice thing about developing this sort of monoclonal platform in this way is it really has allowed a lot of researchers to quickly get their monoclonal sequences, because this is really the quickest way to go from these PBMCs or from the animal to the actual sequence of the monoclonal antibody. We can do it in about 2 weeks, which is incredibly fast, all things considered, and it's very cost effective. So this process does not require any sort of like really high-end machinery. It does require some expertise on the flow cytometry, but doesn't require any sort of high-end machinery and doesn't require these more broadly applied immune bacteriophage library creation and panning or yeast display systems that kind of have been more of the standard in the field for this sort of discovery approach. So in the end, this really is a unique offering that Lampire is in position to deliver on. And this is primarily because of this kind of, dare I say, marriage of the animal work that we are known for and have been experts for many years, and then putting that together with this sort of downstream technology. And so there's kind of this seamless integration and crosstalk, and that's what it takes to get these processes to work robustly and efficiently, is to have those sorts of almost disparate expertise constantly talk to one another.
Oh, very interesting. And going back to your earlier comment about making it through the challenges and celebrating the successes, I'd love to talk about both some of the challenges and some of the successes that you've encountered along your career path. And perhaps we'll start with challenges. I know failures are part of science, right? So do you have a story of a failure or a major challenge, John, that you've had to work through that you can kind of talk us through and explain how you got through a difficult time?
One story would be, and it's not that astounding, but for us, it was a major challenge. At the time, this goes back now several years, when I was working at Merck, and we were looking at new therapeutics in the neuropathic pain area. And one of the targets, and something that it's been disclosed that Merck's works on it, so it's okay if I talk about it, is called NaV1.7. Vertex, I don't know if you know the biotech, but they have a leading molecule in this space right now that is showing a lot of clinical promise for neuropathic pain. And what's great about these NaV1.7 or even NaV1.8 is that they're potentially non-addictive, nothing like an opioid, but have the promise of being very efficacious for cancer-related pains or other neuropathic-related pains, and not cross the blood-brain barrier and not have the issues seen in other sort of pain medications. So these sodium channels or these NaVs, these NaV1.7 and 1.8, there's a big family of these NaVs that are expressed on neurons. So I forget even all of them. There's like NaV1.2, 1.4, 1.5. 1.5 is an important one in the heart. There's 1.7, 1.8, and there's even other ones. But my primary responsibility at the time was to help develop assays to look at the electrophysical properties of these NaV or sodium channels. So we were trying to express and look at the selectivity of our small molecule compounds across all of these different NaVs and across all of the different sort of translational species that we would potentially be studying them in the lab. And there was this particular one, I won't forget it. It was called rat, for the murine species rat, NaV1.4. So we couldn't get this thing to be functional. We were able to express it and actually make a rat NaV1.4 protein, but it wasn't functional for some reason. And we didn't know why. We really honestly had no reason why. And in the world of proteins of these sorts of targets, there's all this sequence information, publicly available through NCBI in a database called GenBank. And again, most, if not every academic researcher that would be doing drug discovery or this sort of work is familiar with GenBank. And as it turns out, GenBank is not always correct.
Oh, no.
They are correct vast majority of the time. And if you go in there and you ask, okay, what's the sequence of some human protein or some mouse protein, they're going to be right. They have it. For these less often used species, they're not always correct. And they specify, some of the sequences are not fully validated. But this was one that was really validated as per their guidelines. But as it turned out, there was one single amino acid out of 1,000 that was wrong. And actually, that was a critical one for its function. So I got some heat because that was my job at the time to be able to make it work. And I believe I just was pouring my heart out, looking where we do a lot of sort of cross-species analysis at the sequence level. And ultimately, I think I saw this one residue and I did some additional structural analysis and we identified, well, maybe this is wrong. And we corrected it and it actually solved our problem. And we made the functional rat NaV1.4 and it worked ultimately.
I'm glad the story had a happy ending. I was like riding the roller coaster of emotions with you. Well, I think making it through these difficult times can be so rewarding. I think if it works all of the time, the first time, it doesn't feel like you've kind of invested as much into it. But I think celebrating these big wins and even these small victories in the lab is tremendously important in science because there's a lot of challenge and failure. So, John, do you have a favorite success story that you'd like to share with us?
Sure. Yeah. So ultimately for me and my teams that I've been involved with, success stories come when you can deliver something that either makes it to the clinic, that means, clinical phase trials, or ultimately get to the marketplace where you can really affect disease positive, let's say, in the broader population. So whenever you have the opportunity to deliver something, let's say an antibody or a small molecule that makes it into the clinic, that's a real success for us. And it doesn't always happen. A lot of our projects fail. But a recent success story, when I was at Molecular Templates not long ago, we're working on these what are called immunotoxins. And these are similar to antibody drug conjugates, if you might be familiar with them. Antibody drug conjugates are really an antibody that has a payload that would be directed to a cancer cell. This would be a more targeted approach versus something like chemotherapy or radiation treatments. This is really kind of combining the best of both worlds of an antibody and, let's say, a radioisotope or some sort of payload that would kill that cancer cell specifically. So we're working on these immunotoxins, and we have an immunotoxin that I was lucky to be involved with, which was made through the use of two of these nanobody domains to make what we call a biparatopic molecule. Essentially, it's just two nanobodies that would hit two unique epitopes on a particular cancer-related cell type. So that was a recent success that we were able to, myself and my team, and more broadly, the Molecular Templates company, was able to deliver this molecule that's now in the clinic being explored for therapeutic use in cancer.
I think that is absolutely remarkable. And can you give us a sense of what was the time frame and perhaps how did you celebrate at the end of this when you were able to hand over that sort of finished, shiny product?
You feel like it's never quite finished, right? And it's still going forward, of course. But the time frame was rather quickly through the preclinical studies and the early discovery phase. In this particular case, about 3 to 4 years, I would say. So it does take a good amount of work on the front end discovery. And then, of course, through your translational models of the disease, in this case, cancer that we are looking at. And then, of course, to do all the proper safety toxicology and all that sort of stuff, which takes significant effort as well. At the end, I think we went out for pizza.
Ooh, I like it.
Actually, it probably was barbecue because Molecular Templates was Austin, Texas-based companies. And I think we celebrated with barbecue.
Oh, I love that. I think taking time to celebrate and acknowledge all of the work and effort that goes into these projects is tremendously important. And I know when you're working on such an exciting project, it can be really hard to pull yourself away from the lab. But we like to encourage our listeners to maybe give their lab minds a break and pick up a book, try to read something for fun. So do you have a book that you've really enjoyed, John, that you'd like to recommend for all of us today?
You're not going to like this answer, but I don't read a lot of books. The last book I read was Sapiens, and I enjoyed that book. But honestly, most of my reading is of scientific publications. Sad but true. I'm a huge podcast listener. Two of the podcasts, I should say, that I'm an avid listener of there's this Mindscapes podcast by Sean Carroll, who's a physics person out of Johns Hopkins University. And then also Lex Friedman podcast, who was originally, I guess, and I think still at MIT. And he explores a lot of different technology and any sort of AI related fields as well on his podcast.
Oh, absolutely. I think learning more about these other fields can be really exciting, can even inspire you in your own field to maybe apply some of the things that they talk about. So we'll say that counts. We'll put these resources on our website for our listeners to find. And I know, John, that being a scientist, there are a lot of opportunities to travel. We mentioned that you've, over the course of your career, worked at a lot of different places, but I think you get to travel quite a bit for training, for working with other collaborators, and for going to conferences. So when you think back over all of your scientific travels, is there a particular place that you really enjoyed that you'd like to tell us about?
The one that my wife always brings up is our trip to Madrid. And this is going back several years now. And I think it was a Society for Neuroscience conference that was happening in Madrid, where scientists from all over the world were gathering. And And I was lucky enough to be able to travel with my wife there. And a couple of the highlights were when we got there, we actually stayed at I would say a really nice hotel. It was one of those scenarios where you go into the hotel and your hotel room wasn't ready. Then at some point it became ready. We went up to the hotel room and the floors were all still wet and it was not really ready. And so we were like, oh, the room's not really ready. And they ultimately put us into this like gorgeous suite. So during the entire conference, we were enjoying ourselves in this beautiful intercontinental suite there. But then more so, just the culture and the sights of Madrid. And I had never been to Spain previous to that. And we explored Madrid, which is a beautiful city, but then also the countryside, which is really beautiful, and the old castles and the nature that surrounds Spain. And I would say the other was when more recently, and this was just this past fall, it was an antibodies specific conference in Paris that was really nice and well done. And again, I was able to travel with my daughter, Emma, who's a young scientist. I'm not a young scientist anymore, but my daughter is. And she knows French. So she basically served as my interpreter while we were there.
I think that's amazing. I think knowing the language can help tremendously. And I was curious, you mentioned you hadn't been to Spain before. Did you know any Spanish, or were you able to sort of pick it up while you were there?
I took Spanish in high school, but beyond that, no. But we got along just fine. Most everybody in Europe knows at least some English.
Yeah, and I think the modern translation tools have become increasingly useful, so you're not sort of flipping through the little book.
True.
Well, these sound like amazing travel experiences. And I think travel is something that a lot of people don't necessarily associate with careers in science. And I think there are still a lot of stereotypes out there about what science careers are like and what scientists are like. And I think it's often not the way that is portrayed in the media, right? Like a lot of the scientists I know are these creative, amazing, funny people. And that's not how most of the public sees us, I think. So do you have examples or stories, perhaps, of things that you've experienced in science that really show this human side that a lot of people don't get to see?
I have a story, but as you just described, it's true. I mean, if you're in a science career, it's really a luxury, I would say, and the benefit of being able to work with people from all over the world. I've been able to work with people in the sciences either directly or through collaborations. And as we just discussed, going to conferences over in Europe or in Asia or more exotic places does really open your horizons. And you're right, that's not really captured very well in what kind of is portrayed as what a scientist is about and what their life is like. One of the funny memories that I have, and this is going back some time, is again going back to when I was at Rutgers. And I'm going to date myself, was back in the mid to late 90s. And this was in the early days of everybody still at home had dial-up connections, whereas like at the universities, we all had high-speed internet, which was great because it did allow us to transfer data. We had networks in the labs and we were doing a lot of data analysis, and we had connected a lot of the laboratories that way. And we had old Macintosh computers in the laboratory, which we would analyze our data and connect to our instruments. But again, this was when broadband connectivity just really wasn't standard. Most everybody had dial-up. And so me and one particular other colleague of mine, we were still young and we liked video games. And we took it upon ourselves to install a version of a video game called Quake, like a role-playing first-person strategy game. And we installed it in one of our lab computers without anybody knowing. Of course, our lab head, Isaac, didn't know at the time, or nobody else, I don't think really knew at the time. So basically, we were running a Quake server out of the lab, probably one of the first Quake servers.
Oh, that's awesome.
And from home, we would dial in and we'd have nice broadband connection to play on our Quake server. This was going back about 30 years ago when this was kind of unheard of.
I think that's amazing. So were you an excellent Quake player?
It didn't last long. It was fun. And actually later on in graduate school, I learned that you can't do that. In fact, one of my other mentors in my graduate study is like, you have to make a decision. Either you continue playing these games or you put your time towards what you really want to do in your life. And I chose the latter.
Well, it sounds like you had a fantastic time there in those early lab days. And I think you're absolutely right. Getting this group together of fun people who are just curious about a lot of different things and interested in a lot of different things can result in some fantastic memories. So
absolutely.
And I know working with fantastic people allows you to solve some difficult problems in science. And you are, of course, working on some very challenging aspects of the field. So if we took away, John, the barriers that normally hold you back, whether that be funding or staff or technology, time, feasibility, whatever the barriers may be right now, is there one question or problem that you would want to tackle most?
From an antibody therapeutic perspective, particular to the use of antibody therapeutics, which can be very safe and effective, is to be able to apply them more effectively to neurological diseases. And one of the major challenges for using antibodies for neurological diseases is proper delivery of the antibody. Because if you take an antibody therapeutic, it's very hard, generally because of the size of an antibody, and it's a protein. And for good reason, you have a blood-brain barrier that restricts what actually gets into your central nervous system or into your brain. So one of the challenges, which continues to be a major challenge, is being able to effectively deliver antibody therapeutics into the central nervous system to affect neuropsychiatric or neurodegenerative related disorders. And I feel like there still is opportunity there. There's definitely a lot of research and discovery in this space, but that is one area where I feel that without any sort of limitations, an area that could really, if we're able to kind of get over that hurdle, then there's a whole lot of opportunity there to affect these sort of neurological related disorders.
Absolutely. I think this is an area of tremendous potential, and we appreciate you sharing this dream question with us today. And I'd love to end our conversation, John, by talking about advice. So for our listeners out there who are likely on their own scientific journeys or journeys through other endeavors in life, is there one piece of advice that you were given that really helped you get to where you are that you can share with all of our listeners today?
It was a piece of advice, but it's also something that turned into something that I learned. And that is, don't be afraid to fail, especially when you're early in your career in graduate studies. You're going to fail. Things are not going to work, but take that as an opportunity. There's always opportunities and failures. You always have learned something that you can apply and ultimately get to where it's a success.
Oh, I love this message to sort of find that silver lining or the lesson to be learned in failures. And I think you're absolutely right. Is there any other last message, or perhaps, John, a last note of inspiration that you would like to leave everybody with today?
I would say, don't be afraid of something new, whether that be a new career, a new direction in your work, a new place of work. I've always was rightfully anxious or maybe sometimes afraid to move to a new department or a new group or new team or new company. But whenever I did, always met new people, always learned something. And it really made my career fantastic. That's what I would say is don't be afraid of something new or somewhere new.
I love that message. And I know, John, today we only scratched the surface of the outstanding work that you are doing there at Lampire Biological Laboratories and your career as a whole. So if our listeners want to learn more, what is the best way for them to do so?
Best way would be to go to lampire.com, which is our website. Through that website, you could get directly in contact with me if you so wanted to.
Phenomenal. Well, listeners, definitely check out that website. Reach out to John if you have any specific questions. And John, it's been such a pleasure to chat with you today. Thank you so much for your time.
Thank you.
Well, it's been a pleasure to chat with you, John. And listeners, wonderful to have you here with us as well. We hope you join us again next time for another episode of People Behind the Science.